DPP HIV 1 and 2. Instructions for oral fluid. The DPP HIV 1 and 2 assay for oral fluids contains the following components: the DPP test device, an oral fluid collection swab, a black cap sample tainer for diluting oral fluid specimens, and a green cap running buffer bottle. Note that the DPP test device has two colored lines in the test window. One is blue and the other is green. Materials required but not provided include a clock, watch, or other timing device. Remove the DPP test device from the foil pouch and place it on a flat surface. Label the device with a patient identification number. Label the black cap sample tainer with the patient ID number. Then remove the top by unscrewing the white cap from the bottle. Obtain an oral fluid specimen by gently swabbing between the upper and lower cheek and gums. Swab 4 times in a circular manner for a total of 15 to 30 seconds. Insert the oral fluid collection swab into the sample tainer and ensure that the swab rests on the bottom of the bottle. Gently break the swab by applying pressure to the shaft at the break notch. Discard the swab shaft into a waste container. Place the white cap with the upper black cap back onto the sample tainer containing the swab tip. After securing the cap tightly, gently shake the bottle back and forth for 10 seconds. The sample is now ready for testing. Remember to mark the bottle with a patient ID. Applying the sample. Before you apply the sample, make sure the DPP HIV 1 and 2 test is placed on a flat surface. Now, unscrew the upper black cap, keeping the white cap screwed onto the sample tainer. Hold it in a vertical position, but upside down, and gently squeeze exactly 2 drops of the sample into the round sample well, without touching the bottom of the well with the tip. Be aware that holding the bottle sideways will produce invalid results. Replace the black cap on the sample tainer vial so the sample is available for repeat testing if needed. Now, wait 5 minutes. Applying the buffer. Ensure that the colored lines have disappeared from the test window before you apply the buffer. Then remove the green cap of the running buffer bottle. Hold it in a vertical position, but upside down, and gently apply 4 drops of buffer onto the square buffer well, again without touching the bottom of the well with the tip. Be aware that holding the bottle sideways will produce invalid results. Replace the green cap after dispensing the buffer into square buffer well 2. After about 3 minutes, a reddish color will spread across the strip in the test window as a signal that the test is starting to run. Read the result. Wait at least 25 minutes, but no longer than 40 minutes before reading the results. And always follow CDC guidelines about how to best inform the test subject. Reading the results earlier than 25 minutes and later than 40 minutes after the addition of running buffer to buffer well may yield erroneous results. First, make sure that a reddish control line has formed above the C marking in the test window. If no control line is visible, then the test is invalid and a new test must be run. If no line has formed above the T marking in the test window, the result is non-reactive, meaning that the patient is preliminarily negative for HIV-1 and/or HIV-2 antibodies. Be aware that this does not necessarily exclude HIV infection. If a reddish line is visible above the T marking and a line is visible above the C marking in the test window, then the result is reactive, meaning that the patient is preliminarily positive for HIV-1 and/or HIV-2 antibodies. Be aware that the presence of any reddish color in the test line above the T marking, along with the visible control line, is considered reactive. Finally, if a reddish control line has not formed above the C marking in the test window, regardless of whether or not a test line is formed above the T marking in the test window, then the test is invalid and a new test must be performed.